Hi, hello, welcome to Mass Physics-Mnr. This is Ramakrishna Zoology faculty. So, in the previous class, we are discussed about what is some genetical disorders in the principles of inheritance and variation. Already two classes we are discussed. Now we will go to third class in this principles of inheritance and variation. Okay, so now in this third class, we will discuss remaining genetical disorders. After completion of the genetical disorders, next we will discuss the pedigree analysis. Okay, so next go to, so in the previous class we are discussed about what is up to thalassemia, that is autosomal recessive disorder. Okay, thalassemia, autosomal recessive disorders. Okay, next go to third disorder in this, that is the phenylketonuria. 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 Okay, so the phenylketonuria affected genes, the affected genes present on 12th pair of autosomes. 12th pair of autosomes. Actually, it is the complete metabolic disorder by the defect of the particular gene from the 12th pair of chromosome. That is, the person is suffered by this little some metabolic disorders, right? So now we will discuss actual function. Phenylketonuria means the presence of phenylpyruvic acids in the urine. Okay, the presence of phenylketone bodies or the presence of phenylpyruvic acid within the urine, that is called phenylketonuria. So what is the reason for accumulation of phenylpyruvic acid within the human blood? Okay, then what effects will be appear in the human being by the accumulation of phenylalanine amino acid within the human being? So, right, come to the point. That is, the phenylketonuria means phenylketonuria. Phenylketonuria means, okay, what is the actual meaning? Urine plus phenylketone bodies. Phenylketone bodies. Okay, it is phenylketone bodies. Phenylketone bodies, nothing but phenylpyruvic acid. This is the phenylketonuria. That is the phenylketonuria. See, actual what is the function of this means the effect, it is caused by the effects of synthesizing the particular enzyme, effects of phenylalanine hydroxylase enzyme, phenylalanine hydroxylase enzyme, hydroxylase enzyme, it is the main thing. So it is very important point. Actually, first we will discuss the functions of the phenylalanine hydroxylase enzyme in the human body. Okay, first the phenylalanine is present in our daily taken food material. Phenylalanine. Okay, phenylalanine. It is one of the amino acid. Clear. This amino acid is generally present in our daily taking food material. Okay, see, now this phenylalanine, okay, phenylalanine, which is after releasing into the blood, okay, which is converted into tyrosine, which is converted into tyrosine. Okay, so it is another kind of amino acid. What is the role of this phenylalanine hydroxylase enzyme role? Means that enzyme is essential for conversion of phenylalanine into tyrosine. Actually, the phenylalanine is the non-metabolic material. Non-metabolic material mean nothing but it is a non-metabolic amino acid simply. Okay, non-metabolic amino acid it is. Non-metabolic amino acid and which is converted into metabolic. It is the metabolic error disease. Actually, this phenylketonuria is the metabolic error disease. It is converted into the metabolic. Okay, now the tyrosine is easily undergoes to digest with the help of certain other products, other enzymes inside the body. It is easily enter into the cell, and within the cell it is easily undergoes to cell respiration for the production of energy. Right, this is the normal function. The presence of what is phenylalanine hydroxylase. Phenylalanine hydroxylase. Phenylalanine hydroxylase. Okay, this is the enzyme. The presence of this enzyme which is converted into this amino acid, now it is easily involved in the it is easily involved in the cell respiration. It is easily involved in the in cell metabolism. That's enough. It is easily involved in the cell metabolism. This is then but for the synthesizing these enzyme, okay, the particular genes are present on the 12th pair of chromosome. On the 12th pair of autosome, what gene is responsible for phenylalanine hydroxylase enzyme synthesizing? That enzyme is present, that gene is present on the 12th pair of chromosome. So, unfortunately, by the occurring the mutation on the 12th pair of chromosome, this phenylalanine hydroxylase enzyme cannot synthesize into our body, within our body. Without this enzyme, the phenylalanine amino acid will not be converted into tyrosine. Understood? See, then what happened? This phenylalanine is still accumulate within the body. Okay. So now by the occurring the mutation, by the mutation on the 12th pair of chromosome, what happened? 12th pair of chromosome, okay, that gene become inactivated. Gene becomes inactivated. Thereby the enzyme will not product. Enzyme will not produce. So without this enzyme, this phenylalanine cannot be converted into this. It is still accumulated within the human body as a phenyl pyruvic acid. Okay, now phenylalanine is there. Phenylalanine. Okay, so now it is without the absence of phenylalanine hydroxylase enzyme. Phenylalanine hydroxylase enzyme. This phenylalanine is accumulated within the body. It is accumulated. It is accumulated in the blood. It is accumulated within the blood as phenylpyruvic acid. As phenylpyruvic acid. Phenylpyruvic. Phenylpyruvic. As phenylpyruvic acid. It is also called with phenylketone bodies. In the high concentration condition, it is the phenylketone bodies. That's enough. So these phenylketone bodies have high concentration, which have a high concentration nature. High concentration nature. High concentration nature. Thereby what happened here? So the accumulation of more pyruvic acid within the blood, when the blood is filtered within the kidney along with the urine, what happened? The phenylketone bodies are also released out along with the urine. Means the presence of phenylketone bodies within the urine, that condition is called phenylketonuria. PKU, that is called, it leads to phenylketonuria. So what are the symptoms? Symptoms of phenylketonuria. Symptoms of phenylketonuria. Symptoms of phenylketonuria. First thing is the mental retardness. Mental retard means mentally retarded, means mentally imbalance, unable to think properly. Next, pigmentation, loss of pigmentation. Okay, loss of pigmentation, right? Loss of pigmentation. Loss of pigmentation. Okay, it is one. Next, what is change? Then pale color of skin. Pale color. Skin pale in color, and also white hair. White hair. Actually, it is this gene is responsible for the pleiotropy. It is responsible for the pleiotropy. Okay, means it is one gene can express more than one character, phenotypic character. So what gene is present on the 12th pair of chromosome that can express more phenotype, more than one, means the same gene is responsible for the what is development of central nervous system. The same gene is responsible for the maintenance of skin pigmentation. The same gene is responsible for the what is maintenance of hair color, skin color. Melanocytes are maintained by the genes only. That's why it is the pleiotropy nature. So that's why the gene is also called as a pleiotropy gene. The gene is also called as a pleiotropy gene. So this is the phenylketonuria. This is the phenylketonuria. So the affected genes are present on 12th pair of chromosome. See, this is phenylketonuria. Affected genes are present on 12th pair of autosome, and phenylketonuria means the presence of phenylketone bodies within the urine. It is effects of phenylalanine hydroxylase enzyme in the human body. For synthesizing these enzyme, what gene is present on the 12th pair of chromosome? That gene is responsible for production of this enzyme. ఒకవేళ ఈ జీన్ మ్యుటేషన్ లో రిమూవ్ అయిపోతే ఆర్ ఇనాక్టివ్ అయిపోతే ఈ ఎంజైమ్ ప్రొడ్యూస్ కాదు. ఈ ఎంజైమ్ ప్రొడ్యూస్ కాకపోతే అసలు ఈ ఎంజైమ్ వల్ల అడ్వాంటేజ్ ఏంటంటే ఫినైల్ అలనైన్ అనే ఒక నాన్ మెటబాలిక్ అమైనో యాసిడ్ ఈ ఎంజైమ్ వల్ల ఏమవుతుందంటే మెటబాలిక్ మెటీరియల్గా టైరోసిన్గా కన్వర్ట్ అవుతుంది. ఈ టైరోసిన్ సెల్ రెస్పిరేషన్లో ఇన్వాల్వ్ అయ్యి ఎనర్జీ ప్రొడక్షన్లో యూజ్ అవుతుంది. కానీ ఇప్పుడు ఈ 12th పెయిర్ ఆఫ్ క్రోమోజోమ్ మీద ఉండే జీన్లో మ్యుటేషన్ జరగడం వల్ల ఏ జీన్ అయితే ఈ ఎంజైమ్ ప్రొడక్షన్లో యూజ్ అవుతుందో ఆ జీన్ మ్యుటేషన్ వల్ల ఇనాక్టివ్ అయిపోతే ఈ ఎంజైమ్ ఆ హ్యూమన్ బీయింగ్లో ప్రొడ్యూస్ కాదు. ఈ ఎంజైమ్ లేకపోతే ఫినైల్ అలనైన్ అనే అమైనో యాసిడ్ టైరోసిన్గా కన్వర్ట్ కాదు. సో మనం తీసుకునే డైట్లో ఇది ఎక్కువగా ఉంటుంది జనరల్గా. ఇది గ్రాడ్యువల్గా మన బ్లడ్లో ఏమవుతుంది, గ్రాడ్యువల్గా అక్యుములేట్ అవడం వల్ల అక్యుములేట్ అవడం వల్ల దీని కాన్సన్ట్రేషన్ మన బ్లడ్లో పెరుగుతుంది. బ్లడ్లో కాన్సన్ట్రేషన్ పెరగడం వల్ల ఫినైల్ పైరువిక్ యాసిడ్గా కన్వర్ట్ అవుతుంది. యాసిడ్ అంటే ఏంటి, హై కాన్సన్ట్రేషన్ కదా. దీనివల్ల బ్లడ్ కాన్సన్ట్రేషన్ పెరిగి మెటబాలిక్ ఎఫెక్ట్స్ అంటే ఇంటెస్టైన్లో డైజెస్ట్ అయిన ఫుడ్ బ్లడ్లోకి అబ్జర్వ్ కాదు. ఎందుకంటే బ్లడ్ కాన్సన్ట్రేషన్ అనేది ఇంటెస్టైనల్ ఫ్లూయిడ్ కాన్సన్ట్రేషన్ కంటే ఎక్కువగా ఉంటుంది. జనరల్గా అయాన్స్ మినరల్స్ అనేవి హై కాన్సన్ట్రేషన్ టు లో కాన్సన్ట్రేషన్ వెళ్తాయి కానీ లో కాన్సన్ట్రేషన్ టు హై కాన్సన్ట్రేషన్ వెళ్ళవు. ఇప్పుడు బ్లడ్లో హై కాన్సన్ట్రేషన్ ఉంటుంది, ఇప్పుడు ఇంటెస్టైన్లో లో కాన్సన్ట్రేషన్. సో ఈ డిఫ్యూజన్ ప్రాసెస్ డిలే అవుతుంది. సో ఇది దీంట్లో ఉండే ఇది ఎక్కువగా బ్లడ్లో అక్యుములేట్ అవ్వడం వల్ల బ్లడ్ కిడ్నీలో ఫిల్టర్ అవుతూ ఉన్నప్పుడు ఆ టైంలోనే ఈ కీటోన్ బాడీస్ కూడా యూరిన్లోకి వెళ్ళిపోతాయి ఫిల్టర్ అయ్యి. సో ఈ కండిషన్, ద presence of phenylketones bodies within the urine, that condition is the phenylketonuria. సో ఈ పర్సన్లో వచ్చే సిమ్టమ్స్ డిజార్డర్స్ ఏంటి అంటే ఫస్ట్ మెంటల్ రిటార్డ్నెస్, అంటే సెంట్రల్ నర్వస్ సిస్టమ్ అనేది ఎఫెక్ట్ అవుతుంది. మెంటల్ రిటార్డ్నెస్ ఎలా వస్తుంది అంటే ఎఫెక్ట్స్ ఆఫ్ సెంట్రల్ నర్వస్ సిస్టమ్, ఇది ఫస్ట్ ఎఫెక్ట్స్ ఆఫ్ సెంట్రల్ నర్వస్ సిస్టమ్. అండర్స్టూడ్, ఎఫెక్ట్స్ ఆఫ్ సెంట్రల్ నర్వస్ సిస్టమ్. దానివల్ల మెంటల్ రిటార్డ్నెస్, పిగ్మెంటేషన్, మెలానోసైట్స్ అనేవి మెలానిన్ పిగ్మెంట్ని సింథసైజ్ చేయకపోవడము, హెయిర్ కలర్ తగ్గిపోవడము, స్కిన్ కలర్ పోవడము, ఇలా. సో ఈ కండిషన్ అన్నిటిని మనం ప్లియోట్రోపిక్గా కన్సిడర్ చేస్తారు. ఇప్పుడు ఏ జీన్ అయితే వీటన్నిటికి రెస్పాన్సిబుల్గా ఉందో ఆ జీన్ని ప్లియోట్రోపిక్ జీన్గా కన్సిడర్ చేస్తారు. అండర్స్టూడ్. సో ఇది ఫినైల్ కీటోన్యూరియా ఎక్స్ప్లెనేషన్. And after that, one more autosomal recessive disorder, that is the cystic fibrosis. It is also one of the autosomal recessive disorder. It is one of the autosomal recessive disorder. That's enough. It is after this, what is the affected genes are present on affected genes are present on 7th pair of autosomes. It is the affected genes are present on 7th pair of autosomes. So what are the characters of this disease? That is effects of exocrine gland ducts or effects of exocrine glands. Effects of exocrine glands and ducts also. Along with this, pancreas and lungs and liver. Pancreas and intestine, inner lining of intestine, inner lining of intestine. So ఇవన్నీ ఎఫెక్ట్ అవుతాయి. సో ఎందుకు అంటే రీజన్ ఏంటి? what is the reason for effect of these all organs? Means excess production of mucus, excess production of mucus, excess production of mucus, ఇది మెయిన్. సో ఇది దీన్ని మనం అండర్లైన్ చేయాలి. మ్యూకస్ ఈజ్ ఎక్సెస్ ప్రొడక్షన్. యాక్చువల్గా whatever cystic fibrosis means what here? The presence of cysts on the pancreas. Generally electrolytes అనేవి మెయిన్గా ఎక్కడి నుంచి ప్రొడ్యూస్ అవుతాయి అంటే ఇంటెస్టైన్లోకి ప్యాంక్రియాటిక్ జ్యూస్ నుంచి వచ్చేస్తాయి. ప్యాంక్రియాస్ ప్రొడ్యూస్ చేస్తాయి ఎస్ఎన్ఏ సెల్స్ అలాంగ్ విత్ ప్యాంక్రియాటిక్ ఎంజైమ్స్. సో దీంట్లో పొటాషియం అండ్ సోడియం అండ్ క్లోరైడ్ ఎక్సెట్రా చాలా ఎక్కువ ఇవంటాయి ఇవి. ఇప్పుడు జనరల్గా ఎస్ఎన్ఐ సెల్స్లో సిస్ట్ ఫామ్ అవ్వడం అంటే ఏంటి? ఇంక్రీజింగ్ ద నెంబర్ ఆఫ్ ఎస్ఎన్ఐ సెల్స్. ఇప్పుడు నెంబర్ ఆఫ్ సెల్స్ ఇంక్రీజ్ అవుతే వాటి ప్రొడక్షన్ కూడా ఎక్కువ అవుతుంది కదా. ఓకే, ప్రొడక్షన్ ఎక్కువ అవుతుందంటే ఎక్సెస్ సెక్రేషన్ ఆఫ్ దీస్ ఎలక్ట్రోలైట్స్ కదా. ఈ ఎలక్ట్రోలైట్స్ ఎక్సెస్ సెక్రేషన్ అవడం వల్ల బ్లడ్లోకి వెళ్ళిన తర్వాత మన ఎక్కడైతే మ్యూకస్ లైనింగ్స్ ఉంటాయో క్యావిటీస్ ఉంటాయి కదా, మీన్స్ ఇంటెస్టైనల్ ట్రాక్, రెస్పిరేటరీ ట్రాక్ క్యావిటీస్ ఉన్నాయి కదా, బ్రాంకియల్స్ క్యావిటీస్, ఆల్వియోలై, ఇక్కడ ఉండే మ్యూకస్ లైనింగ్ కోటింగ్ మొత్తం కూడా థిక్ అవుతుంది బై ఎక్సెస్ అక్యుములేషన్ ఆఫ్ దీస్ ఎలక్ట్రోలైట్స్. ఈ థిక్ మ్యూకస్ వల్ల ఏమవుతుందంటే ఆల్వియోలైలో థిక్ మ్యూకస్ ఫామ్ అయింది అనుకోండి, ఇట్ ఈస్ మే ఇంటరప్టింగ్ టు ద ఎక్స్చేంజ్ ఆఫ్ గ్యాసెస్. ఓకేనా? ఇప్పుడు ఇంటరప్షన్ ఆఫ్ గ్యాస్ ఎక్స్చేంజ్ అంటే ఇప్పుడు ఆక్సిజన్, లో ఆక్సిజన్ అనేది ఏమవుతుంది? బ్లడ్లోకి వెళ్తుంది, అప్పుడు ఆక్సిజన్ డెఫిషియన్సీ ఫామ్ అవుతుంది కదా. ఓకేనా? సో కాబట్టి ఇవన్నీ కూడా 1 by 1 ఎఫెక్ట్స్ అనేవి ఇలా వచ్చేస్తూ ఉంటాయి. లివర్ కూడా అంతే, బయల్ జ్యూస్లో ఎఫెక్ట్స్ అనేవి, లంగ్స్, ప్యాంక్రియాస్ అండ్ ఇన్నర్ లైనింగ్ ఆఫ్ ఇంటెస్టైనల్ లైనింగ్, మ్యూకస్ లైనింగ్ థిక్ అయింది అనుకోండి, ఇంటెస్టైన్లో ఉండే మినరల్స్ అండ్ వాట్ ఈజ్ మనకి ఎలక్ట్రోలైట్స్ అవన్నీ ఏమవుతాయి? డైరెక్ట్గా బ్లడ్లోకి అబ్జర్ప్షన్ జరగదు, మైక్రోవేవ్ తోటి అబ్జర్ప్షన్ జరగవు. దానివల్ల మన ఫుడ్ మెటీరియల్, డైజెస్ట్ అయిన ఫుడ్ మెటీరియల్ బ్లడ్లోకి వెళ్ళదు థిక్ మ్యూకస్ వల్ల. కాబట్టి ఈ కండిషన్ మొత్తం కూడా ఒక జీన్, దట్ వాట్ జీన్ ఈస్ ప్రజెంట్ ఆన్ ద 7th pair of autosome, that gene is responsible for maintenance of the pancreatic cells for the production of normal pancreatic juice, and that gene is maintain the proper controlling of these electrolytes. Understood? So this is the cystic. Fibrous means what? Cysts. Cystic fibrosis means some tumor like structures are found on the pancreas, like that. So it is also what is genetic disorder, and it is also can be transferred from one generation to next generation. It is the what is cystic autosomal recessive. And after that, next we'll discuss one more disease, that is the alkaptonuria. It is the alkaptonuria. Okay, so alkaptonuria is one of the autosomal recessive disorder. It is also one of the autosomal recessive disorder. Autosomal recessive disorder it is. And in the total genetical disorder very important point, మొత్తం హ్యూమన్ బీయింగ్ లో ఉండే మొత్తం జెనెటికల్ డిజార్డర్ లో ఫస్ట్ ఐడెంటిఫై చేసిన జెనెటికల్ డిజార్డర్ ఏంటంటే ఆల్కాప్టోన్యూరియా. ఓకే, it is the first discovered genetical disorder. It is the first discovered, it is the first discovered genetical disorder in human being, in human being. Very important. Okay, and also this disease also called as a alkaptonuria. Alkaptonuria. Okay, alkaptonuria means alkaptone, nothing but homogentisic acid. Homogentisic acid plus it is found in urine. Okay, it is found in urine. So it is the alkaptonuria, the presence of alkaptone bodies within the urine, that is the alkapto. And this disease also called as a, this disease also called as a black lung disease. Black lung, it is also called as a black lung disease. Black lung, sorry, black lung, not black lung, black urine disease it is. It is black urine. Sorry, it is the black urine disease. Okay, how the urine converted into black in color? Okay, how the urine converted into blackish in color? That is the question. Okay, right, come to the point. Generally, in our daily taken food material, okay, so many amino acids are there. So many amino acids are available in our daily taken food material. In this amino acid, one of the amino acid, homogentisic acid. Homogentisic. Homogentisic acid. Okay, this homogentisic acid, which is converted into again tyrosine. Okay, means again it is the tyrosine is converted. Okay, it is the metabolic. It is the metabolic product, and it is the non-metabolic product, again same as phenylketone, phenylalanine. It is the non-metabolic material. Okay, for the conversion of this material into this, okay, for the conversion of the homogentisic acid into tyrosine, homogentase enzyme is essential. Homogentase enzyme. Homogentase enzyme is essential. Homogentase enzyme is essential. This enzyme is synthesized by the particular gene. The gene is present on the third pair of autosomes. Okay, so by the effect of the gene on the third pair of chromosome, these enzyme cannot produce into the human body. Without this enzyme, the homogentisic acid cannot be converted into tyrosine. So it is accumulated within the body as a, what is, alkaptone bodies. It is accumulated as a, it is accumulated as a alkaptone bodies. Alkaptone bodies. Alkaptone bodies. See, these alkaptone bodies concentration is increased in the human blood. How the phenylalanine is filtered within the kidney, same these alkaptone bodies are also filtered within the kidney and released out along with the urine. Once the alkaptone bodies are released out from the body to outside along with the urine, within a minutes, what is, these alkaptone bodies are react with atmospheric oxygen molecules. Thereby what happened? Immediately that urine is converted into black in color. That's why it is also called as a black urine disease. That main symptom is the what is after maturation the urine converted into black color. That is the symptom for the alkaptonuria. It is directly appeared once after maturation within 1 or 2 minutes. Whatever urine is there outside, so that will be converted into black color. Reason these alkapto means our homogentic acid is react with oxygen molecules, immediately it will turn into what is black in color. This is the alkaptonuria. It is also autosomal recessive disorder. On the third pair of chromosome, these genes are also present as a recessive trait. So this is one, and along with this, albinism is also one kind of autosomal recessive disorder. Albinism kuda. Albinism ante complete loss of skin pigmentation, pale color, white color. It is the sun, or what is when that person body is exposed to sunlight, then skin burn, and sometimes the skin cancers are also happened by the albinism. And who are suffered by the melanin deficiency, that person iris are also turn into grayish color. Sometimes that eyes are also called as a cat eyes. So that person is not tolerate the high light condition. Okay, so these type of characters are there in the albinism. Okay, these are the some autosomal recessive disorder. See, up to here we are discussed autosomal dominant in Mendelian disorder and autosomal recessive. In autosomal dominant, we have discussed in previous class myotonic dystrophy, and autosomal recessive disorders, sickle cell anemia, thalassemia, phenylketonuria, alkaptonuria, cystic fibrosis, albinism. So many are there, few important. And these are the autosomal recessive disorders up to here. Then after the autosomal recessive disorder, next Mendelian disorders are the allosomal disorders are there. Okay, up to here only we are discussed autosomal what is dominant as well as autosomal recessive. These both are over. Next go to allosomal dominant as well as allosomal recessive. Okay, next go to another disorders, that is allosomal disorders. First I'll keep this heading, allosomal disorders. Allosomal disorders. See, allosomal disorders are also dominant disorders, and recessive disorders are also there. Recessive disorders, dominant disorders and recessive disorders. These two types are there. Dominant disorders and recessive disorders. Okay. So actually మన ఎన్సీఆర్టీ బుక్ లో అల్లోజోమల్ డామినెంట్ డిజార్డర్స్ ని అసలు మెన్షన్ చేయలేదు. సేమ్ టైం వై క్రోమోజోమ్ మీద ఉండే జీన్స్ కొన్ని ఉన్నాయి. పోర్క్యుఫైన్ హెయిర్, హైపర్ట్రైకోసిస్, ఎస్ఆర్వై జీన్స్, టీడీఎఫ్ టెస్టిస్ డిటర్మైన్ ఫ్యాక్టర్స్, ఇవి మాత్రమే డామినేటెడ్ గా ఉంటాయి వై క్రోమోజోమ్ మీద ఉండేవి. But what dominant genes are present on X chromosome, these are not referred. Okay, that's why we don't go to discussion about the dominant traits in allosomal. Only we'll discuss allosomal recessive disorders. Okay, now we'll go to allosomal recessive disorders. Go to allosomal recessive. So in these allosomal recessive disorders, only we'll discuss what genes are present on X chromosome. We don't discuss about Y chromosome, because what genes are present on Y chromosome, these all genes are present as a dominant nature, not in recessive nature. Understood? That's why what genes are present on Y chromosomes, that genes are also called as a holandric genes. Holandric genes. These genes only can be transferred from father to sons only, which are not transferred to father to daughter. because the Y chromosome is not enter into the daughter. Understood? That's why. Okay. So allosomes means one is the X chromosome, second one is the Y chromosome. What genes are present on X chromosome? These are X linked. Okay, these are the X linked. Okay, here X linked traits. Okay, then it is the regarding this, and it is Y linked. Okay, so Y linked genes. Y linked genes only as dominant. Okay, not as recessive. But what genes are present on X chromosome? These are present as recessive. These are present as a recessive condition only. That's why it is X linked recessive disorders. Okay, X linked recessive disorders. Here X linked recessive disorders, some examples are also there. X linked, because these genes are linked with X chromosome only, nullosomes. X and present as a recessive condition. X linked recessive disorders. X linked recessive disorders. X linked recessive disorders. I'll write some X linked recessive disorders diseases. Some examples are there here. Okay, so one is the color blindness. One is the color blindness. It is one. And the second one is the it is one hemophilia. Second one is the hemophilia. Okay, hemophilia it is. Okay, next one is the Duchenne muscular dystrophy. Duchenne muscular dystrophy. Duchenne muscular dystrophy. Okay, it is short form DMD. Short form DMD. Duchenne muscular dystrophy. So these are the X-linked. Means these genes are present. These color blindness responsible genes are present on X chromosome. Hemophilia, what is responsible genes are present on X chromosome, and DMD caused genes are also present on the X chromosome. Okay, so now we will discuss one by one in these X-linked trait disorders. Okay, first our discussion, we should start with X-linked disorder, X-linked recessive disorder, that is color blindness. X-linked recessive disorder, X-linked recessive disorders. X-linked recessive disorder. Okay, so first I want to give one clarity about this. What trait is present on the X chromosome, the trait should transfer to, okay, where mother to what is sons as well as mother to daughters. But in father, one X chromosome, one Y chromosome is there. What genes are present on the father's X chromosome, the trait only transferred to her daughters, not to the sons. Understood? First I'll write one clarity. So mother is there, mother, and father is there. Okay. Okay. So whatever whatever genes are there in the mother X chromosome, okay, the genes are can be transferred to her daughter's son, right? See, right? Okay, so X chromosome contains genes which are transferred to daughter from the mother as well as sons only, sons again. But what genes are there in the father's X chromosome which are only transferred to daughter, which are only transferred to her daughter, not to the his sons. Okay, his daughter only sharing this trait from the father X chromosome. Okay, so it is one clarity. Generally, the X-linked recessive trait affecting nature is also, okay, affecting nature to males and also females, males and females. Okay, so 1/4 percentage in females and 8 percentage affecting nature is there in males. 1/4 percentage in females, very less percentage affecting, very less percentage, but males are more affected than the females. Understood? So not only that single, so what type of traits affecting nature it is? Color blindness, hemophilia and DMD also. These all the type of diseases affecting nature is this only. Okay, in the recessive trait condition, compare with males, females are very less affected because these trait genes are present on X chromosome. Okay, so in female, what is the karyotype? X and X. Male, what is the karyotype? X and Y. So whenever on these two X chromosomes the presence of only recessive trait, in this condition that female will be affected, otherwise the female will be unaffected. But even the presence of what is on the single X chromosome, if the affected trait is there, that males are will be affected. I'll explain with the help of some problems are also there. I'll explain to you. Very important points these are. Okay, first in the this is the brief information regarding this. Okay, next we'll discuss disease wise. First, color blindness. Color blindness. Okay, so color blindness means unable to recognize the colors or inability to recognize the colors. That is, okay, important point this one. Inability to recognize color. Inability to recognize the colors. Recognize the colors. Inability to recognize the colors. Okay, mainly which colors that person is not able to recognize. Okay, mainly three types of colors that person is not able to recognize. Three types of colors. Okay, these are red color, green color, red, green and blue color. Okay, that person is not able to recognize these three colors. And color blindness are also three types are there. Three types of colors color blindness are there. Three types of colors. Three types of color blindness disease are there. Three types of color blindness disease are there. Color blindness diseases. Three types of color blindness diseases are there. Okay, what are that? One is the protonophia. And second one is the deuteronophia. and third one is the Tritonophia. Protonophia, Deuteronophia, Tritonophia. So what is the meaning for Protonophia? What is the meaning for Deuteronophia, like that? Okay. So in this, which person is unable to recognize red and green color? Red and green color. Okay. So unable to recognize. Unable to recognize. And green color only. Unable to recognize. And green and blue color. Green and blue color. Unable to recognize. For example, it is red, green, blue. Only these two, it is the Protonophia. Proto. And these two, Trito. And only these, Deutero. Protonophia, Tritonophia, Deuteronophia. So these are the color blindness. Means here especially based on the cones. Three types of cones are there. Okay, three types of colors recognized cones are there. By the defect of that cones function, that person is suffered by this. Means that these colors recognized genes are present on X chromosome. Understood? These colors recognized genes are present on X chromosome. So by the occurring the mutation on the X chromosome, especially on these genes, that person is not able to recognize these three colors, red, green, and blue colors. So this is one. Then how can this trait can be transferred to the next generation? From parents to offspring, how can it will be transferred? Okay, we'll discuss with the help of some traits here. Okay, for example, here color blindness. Color blindness. Okay, color blindness is there. In this color blindness disease, the traits, how can we write that karyotypes? For example, recessive. First I'll write the dominant. Dominant. So in this color blindness, we'll take the first letter, that is the C. So dominant is the capital C. Okay, it is the capital C. That's enough. And recessive, it is the small c. Or second c. It is indicate for our identification, it is a small c. Okay. See, on the human being karyotype, for example, it is the female. So homozygous dominant, it is homozygous dominant. So homozygous dominant trait, it is normal. Homozygous dominant, it is normal. That is unaffected. Next, on one X chromosome capital C, on one other X chromosome small c, it is heterozygous dominant. Heterozygous dominant. In this heterozygous dominant trait, okay, they are the carriers, that females are the carriers. Carriers are again always normal. Carriers are again always normal. Mean these are only transferred to their offspring, but they are physically normal. Mean there is no appearance of trait this. On these two X chromosome, the presence of two recessive. It is the homozygous recessive disorder, homozygous recessive trait. Homozygous recessive, homozygous recessive trait. It is the affected trait. It is the affected. It is the affected trait. It is the affected trait. Only when on these two X chromosomes, two recessive genes are present, in this condition that females are affected. It is the female version. Next go to male version. Okay. For example, it is the male karyotype. On the single X chromosome, capital C is there. It is the, this male is normal. So we don't explain like homozygous, heterozygous, like, because only one X chromosome is there in the male. And next another trait, so that is small c there on this X chromosome. It is the affected male. That's enough. Affected. That's enough. These are the karyotypes. So how can these traits can be transferred to the next generation? Means, for example, the color blindness female marry a normal male. Okay, then what is the probability of their progeny for color blindness? So total 4 traits are there. 4 types of parents are there for this color blindness. 4 types of parents are there. So what are, who are the 4 types of parents here? Okay, 4 types of parents. 4 types of parents are there. Okay, 4 types of parents. In these 4 types of parents, one type of parents, X and Y type. Okay, first I'll write these karyotypes only. After that I'll mention the genes, means trait on the X chromosomes. And next third one, and next fourth one, right. Now I'll write the traits on this. Okay, okay, right. Okay, see, if it is the capital C, it is the capital C. Okay, and here also capital C, means both parents are normal. Then what is the probability of their progeny? Completely reciprocal now. Okay, reciprocal means now, so color blindness mother and normal father, what is the probability of their offspring? What is the percentage? Now reciprocal, normal mother, color blindness father. Now quite opposite, both parents are affected for color blindness. What is the probability of their progeny? So these are the traits. 4 types of parents are there. Okay, so now we will identify the percentage of color blindness affecting in their offspring by the using of Punnett chart. By the using of Punnett chart, we will identify easily. First, we'll discuss the first one. Okay, we'll discuss the first one here. First one, this one. So it is common, all offsprings are with normal. Okay, all offsprings. Then first we'll substitute these traits in the Punnett chart. Right, on one side female karyotype, on one side male. So this side X capital C Y. Okay, so what is X? X capital C normal. Okay, X capital C and X capital C again normal, and X capital C Y. See, X capital C Y already we are discussing in the previous. So it is also again normal. X capital C Y again normal, means the probability is 0%. Probability is 0%. Okay, 100% of offsprings are with normal. It is the probability. Next, second one. Okay, for example, on one side. Okay, on one side. Okay, so it is X. It is X. This side male, that side female karyotypes. So here female is affected here. Small c, male is unaffected, capital C. Okay, now just you can cross it. This sperm, this ovum, X capital C and X small c. So X capital C small c, it is what? Carrier. Okay, unaffected but carrier. Okay, next X capital C as well as X small c, again unaffected but carrier. Next here X small c Y, it is the affected, and X small c Y, it is also affected. So in this progeny 50% of progeny is affected, 50% of progeny is unaffected. Reason, all daughters are carriers. Carriers are always unaffected, but all sons are affected. All sons are affected here. So in the total 100%, 25 total 50% affected. Only sons are affected here. See, when the mother is affected for color blindness, all sons are affected, but no one daughter is affected. Then all daughters are with carrier only. When the daughters are will be affected, when mother is carrier are affected, if the father is affected only, in this condition the daughters are will be affected. Otherwise there is no chance for affecting the daughters. Okay, it is the what is third one. Next we'll draw this in another slide. Mother is normal. Okay, father is affected. Okay, what is mother is normal, this is the third trait, and father is affected type. So now we will draw this again. It is the capital C as well as it is the capital C, and it is a small c and Y. It is another Punnett chart. So in our examination in 2023 NEET also one question is there. 2022 NEET one question is there based on this 2022 NEET. Okay, see X capital C, X small c, X capital C, X small c means carrier. Carriers are always unaffected. X capital C, right? X capital C and X small c, again carrier. Carrier means again unaffected. And X capital C Y and X capital C Y. So again the sons are the normal. See, when the father is affected, mother is unaffected condition. Understood? All offsprings are normal and 0% is the affecting nature. Okay, it is one trait. And fourth one is there. When mother is affected, along with the father also affected, what will be the probability in their progeny? So then you guess it, all offsprings are will be affected because the recessive trait is there, homozygous recessive trait on a single X chromosome, that recessive trait is there. That's why all offsprings will be affected. So I'll change this Punnett. So small c, small c, it is one gamete, and X small c and Y. So on one side male, on one side female gametes. Okay, next do it what? What is the answer? What is the affecting percentage? X, X, small c, X, small c, Y, X, X, small c, X, small c, Y. 100 percentage of offsprings are affected. 100 percentage of offsprings are affected. Same explanation is applicable to the remaining X-linked recessive disorder. Just we have to change this gene. In hemophilia, we are replacing the C, H only. If capital C, capital H. If small c, small h, we have to substitute. That's enough. Remaining all percentages are same. Remaining all percentages are same. Understood? So this is the color blindness explanation. Okay. After that, next go to second X-linked recessive disorder. That is the hemophilia. That is the hemophilia. That is the hemophilia. Okay, hemophilia, it is the what is hemophilia is a one of the blood clotting deficiency disorder. Okay, in this condition blood clotting may delay by the absence of the some clotting factors. Okay, it is the delay of blood clotting disorder. Okay, continuous loss of blood, so delay the blood clotting, that is hemophilia. Delay the blood clotting mechanism, delay the blood clotting, and this disease is also called as a royal disease. It is also called as a royal disease. What is the reason hemophilia is also called as a royal disease? Means first time this hemophilia identified in a royal family, in Queen Victoria. Queen Victoria. Queen Victoria. Okay, actually she was suffered by the hemophilia. Hemophilia. It means what? Even a small cut, okay, heavy blood continuously loss without clotting. Okay, heavy bleeding, and it is this disease also called as a bleeder's disease. Bleeder's disease. It is also called as a bleeder's disease. It is also called as a bleeder's disease. Understood? So these are the important points regarding this. Okay. So what are the major role for the blood clotting? Okay. Blood clotting factors are important here. Okay. What are blood clotting factors? Blood clotting. Blood clotting factors has major role. So the deficiency of blood clotting factors, deficiency, blood clotting factor deficiency, thereby the cascade reactions will not continue, thereby the blood continuously lost from the injury place. Okay, so it is one, and different types of hemophilia are there. So total 3 types of hemophilias are there. 3 types of hemophilia. Hemophilia. Actually, total how many clotting factors are there? 12 clotting factors are there. Okay, 12 clotting factors are there. By the deficiency of one any one, then we can decide one type of. In this, based on these 3 types here, one is the hemophilia A. One is the hemophilia A disease. Okay, in the world wise 80% of peoples are, okay, who are suffered by the hemophilia, 80 percentage of 100 what is hemophilia disorders? Hemophilia A. It is the deficiency of 8th clotting factor. Okay, that is the anti-hemophilic globulin. Anti-hemophilic globin factor. Anti-hemophilic globin factor. Okay, next second one is the hemophilia B. So 20 percentage in the 100 percentage of hemophilia patients, 20 percentage are with suffered by hemophilia B. It is the deficiency of 9th clotting factor, that is plasma thromboplastin component. Plasma thromboplastin plasma thromboplastin component, PTC. It is also called as a Christmas disease, means Christmas factor deficiency. Christmas factor deficiency. Next, hemophilia C. It is a rare, very rare disease. It is the deficiency of 11th clotting factor, that is plasma thromboplastin antecedent. Plasma thromboplastin antecedent. Plasma thromboplastin antecedent. Actually, the hemophilia C, it is the autosomal disease. It is not related to what is X-linked disease. Only for hemophilia A and hemophilia B, these two are only X-linked related. X-linked. Okay, hemophilia C is it is very rare and autosomal disorder. That what genes are responsible for hemophilia C, the genes are present on the autosomes only. Okay, so these are the hemophilia. Same affecting nature, 1/4 in female and 8% in male. That is applicable to all recessive disorders, means X-linked recessive disorders. Understood? So these are the important points regarding this. So then how can this trait can be transferred from parents to offspring? Okay, now we will discuss the trait, hemophilia trait. Hemophilia trait. What is the hemophilia trait? Same 4 parents are there here. 4 parents here. Parent 1, same mother and father, and second mother, father, and mother and father, and same mother and father. Okay, these are the 4 traits as usually. Same as color blindness trait. కలర్ బ్లైండ్నెస్ లో ఎట్లా చూసినామో సేమ్ ఇక్కడ కూడా అదే కేటగిరీలో చూడాలి. జస్ట్ వీ హ్యావ్ టు చేంజ్ ద జీన్స్ ఓన్లీ. ఇక్కడ ఓన్లీ జీన్స్ మాత్రమే చేంజ్ చేస్తాం కానీ వేరే ఏం చేంజ్ చేయం. పర్సంటేజ్ కూడా ఈ సేమ్ వస్తుంది. కలర్ బ్లైండ్నెస్ దీనికి పర్సంటేజ్ కూడా సేమ్ వస్తుంది. ఓకే, ఐ విల్ చేంజ్. సో ఫస్ట్, వాట్ ఈస్ నార్మల్ మదర్, నార్మల్ ఫాదర్? What is the probability? You can do it with you substitute in the Punnett chart. So like this, already you know very well this. On one side male gametes X capital C Y, on another side female gametes X capital C. Then you can write the percentages. Okay. Next, here also, what is mother is sorry, here it is the hemophilia. Hemophilia means I'll write the first letters of H. Hemophilia first letter is what? H. So it is capital H, again capital H. Capital H is there, nothing but what it is? That is the normal. Capital H is there, that is normal. Small h is there, that is the affected. So here small h, again small h, here capital H, and capital H and small h, and both parents are affected with hemophilia. Small h, small h. Simple, same thing, color blindness, small c, small h, like that. And you can substitute here. Then you can write, type the message, the percentage of this color blindness. Okay, I want to know your understanding level by sending answers. I want that percentages. Male gametes, female gametes. Male small h, y. Female capital H. And same, you can find out it is X small h. Okay, then I want the total percentages. Okay, which parents are having 100% of unaffected, 100% of affected. I want to know this. This is the hemophilia. That's enough. And after this, next third X-linked recessive disorder, that is the DMD, means Duchenne muscular dystrophy. The disease Duchenne. Disease is Duchenne muscular dystrophy. Duchenne muscular dystrophy. Duchenne muscular dystrophy means DMD. DMD. Okay. So it is also one of the X-linked recessive disorder. X-linked recessive disorder. It is also one of the X-linked recessive disorder, that is clear. Okay. See, and it is the muscle disorder. Muscle disease means what are the symptoms here? Programmed death of muscles. Okay, programmed death of muscles. Programmed death of muscles. That's enough. Programmed death of muscles. See, actually in this disease only males are affected. Only males are affected. Okay, no one female affected. No one female affected. Very important point it is. No one female affected. No one female affected. See, then I want to give some explanation regarding this. Myotonic dystrophy is there, na? Myotonic dystrophy, Duchenne muscular dystrophy, these both are different. That is muscle disorder, it is muscle disorder. But that is happening in adulthood age. But it is happened from 4th or 5th year. 4th or 5th year only. 4th or 5th year it started. Once that what is that baby born, then actually why the males are only affected means always for this DMD the females are present as a carrier, and which male is born with this trait. So these symptoms are started at 4th to 5th year, okay, and that person can die at 14 or 15th year age. 15th year age, that persons are died. Before marriage, that males are died. Okay, it means what? So there is no chance for transfer of this trait to the females. Only males are affected by this trait. And see, actually the programmed death, programmed muscle function or muscles are died. Then how? So who born with this homozygous recessive trait? Okay, in this condition, homozygous means what? Mother is carrier, father is normal as usually. Mother carrier means one of the X chromosome has the recessive trait. When the X chromosome recessive trait contain X chromosome bind with Y chromosome, means fertilized with Y chromosome contain sperm, and that zygote will develop into male. So that male, okay, first 4 years that person is present as a normal, or 4th or 5th year almost. From the 5th year that muscle functions are programmed loss. Programmably, what is that muscle functions are gradually collapsed their function. In this process, 10th year, so maximum that person is like a creeping way. That person is not able to walk as well as not able to move properly with the hand also. Finally at the 14th 15th year age creeping, finally the skeletal means that breathing muscles are also lost their function. Once the breathing muscles are stopped their function, that person breathing will be stopped, thereby that person will die. Understood? So that's why this disease and this trait cannot be transferred into the females, but always females are present as a carrier once it is transferred in their parents from by the occurring the mutations actually, not by their parents, because the no one male parent not survive up to marriageable age. Understood? So this is the Duchenne muscular dystrophy. It is started at the early or childhood age. It is started at the childhood age. It is started at the childhood age. So up to here, these are the X-linked recessive disorders. Okay, means up to here these are only Mendelian disorder, defect of gene only. Okay, in the next class we will discuss chromosomal syndromes. Okay, we'll discuss the chromosomal syndromes in the next class, and in the chromosomal syndromes we'll discuss autosomal and allosomal. After that we'll discuss the pedigree analysis. Okay, maximum in the next class we'll complete these zoology genetics topics. Understood, right? And until that you keep watching these videos, and if you have any doubts you type and we will give the reply to you. And also this total notes is we will keep in that videos in that web, means in YouTube, and you can follow it, right? Thank you very much. We'll meet in the next class.